A computational approach for the modified peptides was also developed by the scientist known as John Cates. As we have seen earlier that the databases for the proteins that were unmodified were also developed by the same scientist. So he has also developed the databases, virtual databases that contains the modified peptides. So the spectrum that was generated by the experimental data, they were searched, and in the linear search they were compared with the modified peptides that are present in these virtual databases. But the drawback of this databases and this approach was that despite few of the modifications were used to generate the modified peptides, a large number of combinatorials peptides were generated when we consider a very few number of modifications. So by using this approach, we can determine the modified protein identification problem, and if we want to find a peptide from a database that best matches the experimental spectrum with up to k modifications, then the input of the algorithm will be a database of proteins, means the modified databases of proteins, and experimental spectrum that is generated after experiment of my spectrophotometer, a set of ion types delta, a parent mass M, and a parameter k capping the number of modifications. Because we know that there are more than 200 modifications that could be present in the proteins after translation. So that scientist John used few of the modifications. So output of this algorithm will be a protein of mass M with the best match to spectrum S, that is the most k modifications away from an entry in the database. The major problem with the modified protein identification problem is that whenever we have a similar sequence of two proteins, P1 and P2, but when we do the mass spectrophotometer, they give the very different spectra S1 and S2, and this difference is because of the modifications present in those proteins. So the notion of this solution is that to develop a spectrum that correlates with the sequence of the proteins. But the problem is whenever there are modifications on the similar proteins, the spectra we get is very different from each other. We can overcome this problem by considering the shared peak counts, and we can consider the proteins that are similar with each other. But this problem again arises when these proteins also contains mutation in some of the regions in the amino acids. More are the mutation, the more we get the different spectra. So the modifications that are present on the proteins, they provide or they give the different spectra even when they are similar in the sequence.